Hi, everyone. My name is Frank Wendt. I am a postdoctoral fellow with Renato Polamonti at Yale School of Medicine. Today, we'll be talking about post-traumatic stress disorder and some of the historical studies that have led us from gene discovery to understanding some disease biology in relation to PTSD. I don't have any conflict of interest to disclose. So PTSD is ultimately sort of the end product of a series of events, including pre-trauma risk factors and traumatic events. But in order for us to really understand what's going on with PTSD, we have to have some understanding of what's happening prior to diagnosis of PTSD, including those traumatic events and the pre-trauma risk factors. So looking at PTSD in the DSM-5, I've listed here the criteria that are used for diagnosis, the description of each of those diagnostic criteria, and some example qualifiers for each of those criteria. We see here some of our sort of common themes that we think about with PTSD, including re-experiencing, hyperarousal, and avoidance symptoms. We also see some qualifiers in terms of the length of time these disturbances are happening. This is required for at least 1 month. And we also see some qualifiers here for what the disturbances cannot be attributed to. So in many ways, this qualifier is more of an exclusionary qualifier where we have to make sure that the disturbance is not due to an illicit substance use, some other medication use, or another medical disorder. Looking at some of those traumatic events, we're showing here the lifetime prevalence of those traumas, and those are listed across the x-axis, and they are broken down into a general category of trauma. We see things like accidents and some of these other trauma of a loved one are fairly prevalent in the general population. Overall, across the lifetime, there's approximately a 70% prevalence of any trauma. And we know that a relatively small proportion of those individuals, something like 6 or 7%, go on to actually develop PTSD. So even though most of the population does experience some traumatic event throughout their lifetime, relatively few individuals actually end up with a PTSD diagnosis. We know that many of those traumatic events have some pre-trauma risk factors. Some of the major ones include gender and sex, age of trauma, education, socioeconomic status, and other psychiatric comorbidities. We do know that probably one of the most prominent of these pre-trauma risk factors is gender. We know that women are 4 times more likely to develop PTSD when compared to men when we account for exposure to a traumatic event. And we also know that many of these traumatic events produce the same PTSD risk in both men and women, including accidents, natural disasters, and sudden death of a loved one. However, things like a sexually assaultive trauma have a much different PTSD incidence between sex, where we see that even though women are more likely to experience sexual trauma, we see that men are more likely to develop PTSD following those sexual traumas. So to summarize here, we have 3 timeframes that we're primarily interested with PTSD. We have a pre-trauma, a peri-trauma, and a post-trauma timeframe. And we'll talk about all of these sort of in concert as we move through the next couple of slides. So we'll be walking through quite a bit of material today and sort of touching on each of these topics individually. I've provided here sort of a one-liner for people who are less familiar with this type of study to sort of acclimate themselves and get prepared for what will be shown on each of the slides. If you're familiar with all of these things, feel free to skip over this and move on to some of the actual meat. If any of these topics are unfamiliar to you, please feel free to reference the one-liner provided here. We'll start with twin studies. On the left side of the screen here, we see a really nice summary of some of the early twin studies showing that as the female percentage in the twin study increases, the PTSD heritability estimate also increases. We'll see this trend kind of following through into our GWAS studies of PTSD as well. On the right side of the screen, we see a twin study that attempted to sort of separate out the additive genetic component, the shared environmental component, and the non-shared environmental component between some of those traumatic events, including, in this case, an assaultive trauma category and a non-assaultive trauma category. We also see that many of these twin studies have identified some comorbid conditions in relation to PTSD. In particular, those sort of highlight some other psychiatric diagnoses. We see insomnia pops up quite readily, and some other sleep-related disturbance phenotypes that pop up quite readily. In candidate gene studies, one of the first of which was looking at DRD2, that's the dopamine receptor D2, comparing the A1 and A2 alleles at DRD2, and there was a significant relationship between the hyperreactivity or hyperarousal PTSD subcluster and DRD2 locus. I've also listed some additional candidate genes on the bottom left side of the screen. You'll notice some of the sort of favorite psychiatric disorder players here, including COMT, which is the catechol methyltransferase locus. And then we see on the right side of the screen some of the history of those candidate gene studies and where those p-values lie in some of those additional studies. So moving into genome-wide association studies, these are, this slide and the next slide is pretty much just an introduction to GWAS before we get into what the PTSD data look like. Genome-wide association studies are essentially a brute force experiment that is by nature hypothesis generating. And essentially we are looking at the differences in allele frequency for single nucleotide polymorphisms or single base changes in the DNA between two groups of individuals. In the case for PTSD, we might be looking at a group of individuals who are diagnosed with PTSD versus a group of individuals who are not diagnosed with PTSD. Because we only have two outcomes, this is considered a logistic regression, and this regression model is typically covaried for a number of confounders in these disease association studies, including age, sex, and some principal components of ancestry, essentially the proportion of ancestry assigned to an individual. We also typically covary for a technical artifact. In this case, I've listed batch number here. And the example I'm showing here is a logistic regression with two outcomes, although we could also be looking at a continuous or quantitative outcome. And if we're looking at PTSD, this might be something like the total symptom count from the PTSD checklist. I'm showing here a number of different Manhattan plots, which is the visual representation of GWAS results. We see a number of different patterns here, but all of them have the same structure. So on the x-axis, we have all of the human autosomes. Those are all of the non-sex chromosomes in humans. On the y-axis, we see the -log10 p-value for the association between each data point and our phenotype of interest. Each of those data points represents one of those single nucleotide polymorphisms that I discussed on the previous slide. What's important to note here is that depending on how we define our phenotype, we typically will observe a slightly different pattern in our Manhattan plot. So on the left side of the screen, we're looking at a quantitative versus a binary definition of PTSD. On the right side of the screen, we're looking at stratifying our PTSD data by males and females in separate studies. And we'll see that each of those patterns of our Manhattan plots look slightly different depending on how we've defined the phenotype. On this slide, we're showing how that whole pattern of SNPs or that whole pattern of genetic architecture overlaps with other phenotypes. On the left side of the screen, we see that PTSD shares genetic risk with anthropometric traits, some behavioral phenotypes, some cognitive phenotypes, and certainly some psychiatric disorder phenotypes. On the right side of the screen, we're not necessarily looking anymore at what PTSD is and is not associated with on a genetic perspective, but now we're looking at how does PTSD compare to some other disorders. So PTSD, for example, is associated with a number of anthropometric phenotypes, but when we look at the major depression category on the far right side of the screen, we see that major depression has some genetic correlates that are not observed when we look at the PTSD category, and those typically align with the anthropometric traits, at least in this figure. So one of the major pitfalls to the current status of genetic studies is that there is this overwhelming representation of European ancestry in our genetic studies. And this means that most of our findings are generally only applicable to other individuals of European ancestry. And as you can imagine, this puts a considerable hindrance on applying any of this information to a clinical practice. So to sort of mitigate that downfall or that pitfall of the field, the PTSD genetic research community has really made a push towards understanding how genetic risk for PTSD exists in individuals of admixed ancestry. And this is done by using a method that essentially paints your chromosomes, and we can see that on the bottom left side of the screen where we have red and blue that indicates different part of the chromosomes that are of different ancestries. And then we can perform our genome-wide association studies using this information in our model. So next we'll be talking about gene by environment interactions. Because PTSD relies so heavily on the presence of some index trauma, it's extremely important to consider how different environments interact with risk for PTSD. However, no large study to date actually looks at PTSD as the outcome in terms of gene by environment interaction. There are a number of different studies, however, that look at PTSD or related traumas and pre-trauma risk factors as environments. And one of these will be presented by me on Tuesday, so please feel free to tune in for that between 2:15 and 3:45. Next, we'll look at epigenetics and transcriptomics. This is really important because we can actually get relatively strong effect estimates using slightly smaller sample sizes. This is because these epigenetic and transcriptomic changes can be directly related to either causal mechanisms or downstream consequences of the phenotype of interest. Looking at epigenetics, we see that PTSD is associated with a number of genes that share risk with some other psychiatric disorders, and that's shown in the top right from Smith et al. We also see that some of those PTSD subdomains, in this case avoidance symptoms, are positively correlated with accelerated DNA methylation age. In terms of transcriptomics, we have two types of information shown here. On the left side of the screen, we're showing peripheral tissue, in this case it's blood, and on the right side of the screen we're showing some of these gene expression changes using brain tissue. And both of these studies and sort of this transcriptomics of PTSD in general converge on this concept of a dysregulated inflammatory response in PTSD. And this is really important because it's much easier to study a minimally invasive tissue like blood than it is to study brain tissue. So if we can find things that overlap between those two tissues, we can make some more translatable findings that are hopefully more meaningful and applicable to individuals with PTSD. Finally, looking at neuroimaging, we see two examples here on the left and right side of the screen where we identify hippocampal markers of current PTSD and some of its genetic correlates. And this is a really nice study because it breaks down PTSD into civilian cohorts, military cohorts, and then by sex as well. And we know that some of these things are hypervariable between risk factor, right? Especially the male and female dichotomy in some of these pre-trauma risk factors. On the right side of the screen, we see an association between the putamen volume and PTSD and anxiety disorders. And this is really nice because we're able to see brain regions that sort of confer risk for not only PTSD, but a number of other correlated phenotypes. So to conclude, molecular studies of PTSD and its symptom clusters reveal an extremely complex architecture that overlaps with psychopathology and neurodevelopmental disorders, as well as some psychiatric phenotypes. But also what we're finding is that there's a number of peripheral disorders and even non-disease phenotypes that overlap with PTSD. We see this quite readily with anthropometric phenotypes. We also know that sex, ancestry, and trauma type specific risks are abundant across all of these molecular investigations. And we know that this induces some level of heterogeneity on top of the diagnostic complexity that exists for PTSD already. Sample sizes are increasingly growing, and this is happening quite rapidly. And this will make our studies much more robust, and in particular, allows us to start stratifying the epigenetic, transcriptomic, and neuroimaging studies to make inferences and conclusions about specific types of changes in those heterogeneous cohorts that make up our PTSD cases. Finally, given the stigma associated with some trauma types, it is imperative that both researchers and clinicians engage with both high-risk communities and general communities to begin reducing stigma and improve PTSD scientific literacy and advocacy. And with that, I will provide thank yous to Karison and Caroline and Geeta for their critical feedback, as well as the PGC and the MVP and the WCPG organizing committee. And the contributors of this work are myself, as well as Renato Polamonti. And thank you all for tuning in.